Two types of enzyme. Question 4. Diagram shows how two types of enzyme can be involved in controlling the activity of a protein in response to the presence of a signalling molecule within the cell. Intracellular signalling molecules are often produced as a result of extracellular signals received by cell surface receptors. It's kind of like they've broken signalling into two questions, because you had in question 3 you had hydrophobic, and now we've got hydrophilic. Okay, you've got the intracellular signal molecule is present, which means the target protein, which is inactive, becomes active using a kinase, and then the intracellular molecule is removed, and then an enzyme returns it back to the inactive protein. Okay, explain how the action of protein kinase can switch a target protein from inactive to active. Right, so this definition of what kinases do. So kinase, okay, transfers a phosphate, or you could say just phosphorylates, enolates. Okay, and what you should always be aware of is that this packs a real big punch in terms of what it does to a molecule, and the big one that you want to use, which causes conformational change. Okay, and you need both of that. So you need to say that you've got your change, and then you've got to say it's because of the phosphate that's causing that change. Okay, name the type of enzyme represented by enzyme X. Okay, so we've now got it going back the way. So again, straight KU, kinase adds phosphates, phosphatase removes. That's it. Explain the importance of the system being able to return the target protein to its inactive state. Well, if you didn't have it back in its inactive state, it wouldn't be able to respond. Okay, so basically it returns it to a state it can respond again. In its state, that doesn't make sense. Returns it to a state so it can respond. Okay. Protein kinase A, pKa, is an enzyme that is involved in this type of signalling. To test the hypothesis that pKa is found in a variety of cell types, cell extracts were prepared from different cell types, proteins in the extracts separated by electrophoresis in a gel, proteins were blotted onto a solid support, and an antibody recognising pKa, which is anti-pKa antibody, was used to detect the presence of pKa. Describe how protein electrophoresis is used to separate proteins. Kind of surprised that they've given you 3 lines to say this, because it's pretty straightforward. Okay, they're separated by charge, and that would pretty much being that's your main one. Separated by charge according to their own charge or mass, and that's basically what you're looking for. Okay, explain how the anti-pKa antibody would be used to detect the presence of pKa. Actually, I'm going to move this slightly to the side so I can do a little doodle, if I can get my mouse to move me. There we go. Okay, so if you've got, assuming that we have a little blob of your pKa, then we have our anti-pKa has to come in and attach to it. Okay, now at this point you've no way of knowing that, so your first mark is for saying that the anti-pKa antibody will attach to the pKa. Okay, but then it's saying how would it be used to detect it. The next thing is I need to report somehow, so you need to give an example of how this could be adapted at the top end here, basically, to show you, and your options here are pretty straightforward. Okay, well, there are several that you can go with. The easiest is probably to say fluorescence. Okay, so if you have a fluorescent label, okay, you could go with any other form of coloured label. Okay, and you could go with an enzyme reported. Okay, so the enzyme reporter would mean that you have an enzyme attached up here which you can add the substrate to, and that substrate will then be altered to product, and the product will either be a colour or something that you can detect. Okay, so you need one of those given, and you need to say that it would attach. So first mark is that you've got that the antibody will attach to the pKa, and that labeled antibody, being precise. Okay, and then the label will then be detected, and that's it.